s- Hello everyone, and welcome to our last Abberior Instruments Summer Symposium. My name is Janina Hanne, and I'm the Scientific Relations Manager at Abberior Instruments, and I will guide you through today's program. Today's topic is about life-cell STED microscopy, and I'm super happy that we have 4 great speakers today. First, Francesca Bottanelli and Florian Grimm will give a talk each about life-cell STED microscopy and suitable dyes for it. And then my colleagues Isabel and Martin will show you on our facility line how to acquire life-cell STED images with our adaptive illumination feature. Don't forget to ask any questions that come to your mind in the questions tab of this webinar software already during the presentations. We'll answer as many questions as possible, and we'll follow up on the ones we couldn't answer directly. Now, let's start with our first speaker. Francesca Bottanelli was recently appointed assistant professor at University of Berlin. As a biotechnologist by training, she developed a passion for imaging and cell biology already during her PhD at the University of Leeds. She then moved to the US to carry out a postdoc with James Rothman, where she worked on the development of novel labeling strategies for multicolor life-cell STED imaging in collaboration with the Beversdorf lab. And then she applied these tools to better understand how the Golgi works. In her independent position, she continues to pursue both her passions, STED microscopy and the Golgi. The title of her talk today is Investigating Cellular Logistics with Life-Cell STED. Francesca, I'm really looking forward to your talk. The stage is yours.
Thank you, Janina, for the great introduction for organizing this. And I'm sharing. Can everybody see me and my screen? Yeah. And yeah, thank you for organizing this, and also thank you for to everybody at Abberior for giving me the opportunity of showing you what we are doing with life-cell STED and like how we are using life-cell STED to try to understand intracellular trafficking. And that brings me to my first slide, which is a little bit of an introduction of what we do in the lab. So in the lab, I'm gonna change this to laser pointer. Yeah. So in the lab, we want to understand how proteins get to the right intracellular destination. And in particular, with our work focuses on the Golgi apparatus. The Golgi apparatus, it's a stack of flattened cisternae, and it's often regarded as the post office of the cells because it has really a major central role in sorting to other intracellular organelles. For example, it sorts back to endoplasmic reticulum, or it sorts protein to the endolysosomal system for degradation, or to proteins to the plasma membrane for secretion. And while this is just a schematic representation, all these intracellular transport steps occur over extremely short distances. So this is a textbook electron micrograph, and this is what the Golgi looks like. It's literally a stack of pancakes. And if a vesicle needs to go from the Golgi to the ER, and this is a 1 micron scale bar, that vesicle only will need to travel a few hundred nanometers. And that's why we need super resolution imaging if we want to look at this cellular process. When we image the Golgi with confocal microscope, it's kind of like a blurry haze, and it's only when we use super resolution, in particular STED here, that we start appreciating structures, for example, little vesicles, which are budding off the Golgi. And I structured this short talk into 2 parts. In the first part, I'm going to talk to you about work that I did as a postdoc in the Rothman and Beversdorf labs, where we developed methods for multicolor STED nanoscopy in living cells. And then I'm going to hopefully show you some example and convince you that live-cell STED is being really instrumental to understand the mechanism of membrane trafficking at the Golgi. And just a brief introduction for the few of you that haven't seen the previous webinar and not familiar with STED. So a STED microscope is basically a confocal microscope where depletion donut is overlaid to your excitation beam. And what this depletion donut does, it switches off the fluorescence around the excitation. And if you saturate this depletion donut, your resulting fluorescent imaging is a sub-diffraction size point spread function. So it's around, I would say, 30 nanometers, also in the XY direction, also on commercial STED microscopes. And people have done STED imaging with using fluorescent protein like GFP and YFP. However, we like to use organic dyes because they're much brighter and much more photostable than fluorescent proteins, and that's why we use self-labeling enzymes of the SNAP, like SNAP and Halo tag, to label our protein of interest with our favorite organic dyes for STED microscopy. And the way that it works, that they are enzymes, so they react with a ligand, a benzyl guanine in case of the SNAP, and a chloroalkane in case of the Halo, which is conjugated to a fluorescent dye. And what results from the reaction is that your organic dye becomes covalently attached to SNAP and Halo. And they are orthogonal to each other, so they can be used in multicolor experiments. And talking about dyes, what I really think was a major milestone for live-cell STED was the development of silicon rhodamine back in 2013. So SIR was really the first cell permeable, so that can permeate cellular membranes and can label intracellular targets. The first cell permeable, bright, photostable, steadable organic dye, as it was beautifully showed on the original paper with the microtubules and centrosomes. So we can excite it at around 640 to 650 nanometers and deplete it with the 775 nanometers STED laser, which is equipped on all the commercial microscopes, and it's also our favorite one. And obviously, if you're a cell biology, you want to look at things into a context. So following one single color was really not sufficient for us. So we wanted to bring this to become like a multicolor, to have like multiple color to be able to follow different targets at the same time. And we put some thinking into that, and we thought, so what is the characteristic of the ideal STED dye that we combine with SIR for true color experiments? Obviously, it needs to be bright and photostable. It needs to be cell permeable as well, because we want to label intracellular target. It needs to be depleted by the 775 nanometer laser, so the emission tail needs to be far out enough in the red so we can hit it with that same STED laser, and ideally excited in the red, so like 580, 590 nanometer range. And the reason for this is that at the time that was the favorite hardware combination, that was the favorite combination of color also for fixed cell imaging. So like a red and a far red dye that you can deplete with that same 775 nanometer laser. So just to cut a very long search short, we use silicon rhodamine together with ATO 590, which that has all the characteristics that I discussed in the previous slides for doing true color live-cell STED experiments. And there is actually, in the last few years, there has been like an awful lot of developments, which I think Florian will go into much more detail about. A lot of development in terms of STED, live-cell STED compatible dyes that can all be used with a single 775 nanometer laser. And usually for true colors, people use, as I said, a red dye and a far red dye. But actually, people have already also performed three-color live-cell STED experiments using so-called long stalk shift dyes that can be excited by a green laser and have an emission tail that is far out in the far red. So you can hit it with the same 775 nanometer laser. And this is actually a table from our review that will hopefully be submitted soon about live Cell STED. So watch out for that. And how does it work practically? So you take your cells, you express your fusion proteins. In this example here, we have Sec61 beta, which labels the ER membrane, which is fused to a halo tag, and OMP25, so outer mitochondrial protein 25, which labels the outer mitochondrial membrane fused to a SNAP tag. And the way that it works is actually very simple. You add the dyes to the culture medium, incubate your cells for 1 hour, and then wash out the dye and go to the microscope. And now I'm going to show you some examples of what we did back then to show that this labeling strategy can be used to image any intracellular target of interest or combination of. So, for example, here we have the endoplasmic reticulum and the mitochondria in green, and we can observe events that we wouldn't be able to observe with the standard confocal microscope. So, for example, where I'm pointing down here, there is a mitochondrion that is being constricted by the R. And we can see the hollow center, sort of the hollow lumen of the ER tubules, which are only around 100 nanometers from membrane to membrane. Just another example here, if you focus on the plane of the plasma membrane, you can see, you can follow clathrin-mediated endocytosis events. Here we have labeled clathrin-coated pits with a SNAP tag, and endocytic cargo like transferrin receptor with the halo tag. And we performed our live Cell STED experiments, and you see all these little tiny donuts, which are clathrin-coated pit, wiggling around on the plasma membrane. And for example, down here you see a little clathrin-coated pit, which is filled with cargo that is being endocytosed. And again, these structures are only around 100 nanometer from membrane to membrane. So super resolution is really necessary here. And when you have highly overexpressed protein, you can actually image for quite a while. And in this case, again, to show the power of the technique, we have Halo Sec61 that labels the membranes of the ER, and a SNAP KDL protein that labels the lumen of the ER. And not only you can see the hollow center of these tubules, but you see that they're filled with the cargo, and you see also these tri-junction, which are filled with the SNAP KDL protein. So coming back to our main biological interest, I hope I really convince you in the next 5 to 10 minutes that live Cell STED is really being instrumental for us in trying to understand the mechanism of polarized secretion in polarized epithelial cells. So because that's what the lab is working on. So polarized epithelial cells has different types of plasma membrane. So you have an apical plasma membrane, you have a basolateral plasma membrane, and you have tight junction that separate these two extracellular environment. And the same Golgi, in particular, I'm going to say TGN a lot. That's the trans-Golgi network, which is just the last cistern of the Golgi, which is involved in further sorting. The same Golgi needs to sort to so many different plasma membrane destinations. And it is actually still unclear how it does that at the nanoscale level and in living cells. So one of the limitations of our work is that all these organelles that coordinate sorting, which include the TGN and also recycling endosomes, and the TGN and the recycling endosome are considered the two most important sorting stations for polarized sorting. So all these organelles are tightly packed in the perinuclear area, so in the area around the nucleus. And the schematic here tells you why, again, stresses why we need super resolution imaging. These organelles interact with each other, and they are less than 100 nanometers apart. The different Golgi cisternae are around 20 nanometers apart and only a few tens of nanometers in thickness. So this is really why we need super resolution again. Another thing is that if you are studying traffic, if you're a cell biologist, what is also very important to stress is that all these tubular endosomes and trans-Golgi network elements are very sensitive to fixation. So you fix your cells with 4% paraformaldehyde, or even just adding a little bit of glutaraldehyde, and these structures are completely gone. So that's how our Cell STED labeling strategy became very handy. And additionally, which is a further limitation for our work, is that we work on very important cellular machinery components, and we don't want to overload the cells and overexpress them. So what we do, we actually use CRISPR-Cas9 to endogenously tag, so to tag with SNAP and HALO the endogenous copy of the gene, so that we do not have any overexpression issues, which end up basically masking the phenotype, or like messing up with the phenotype that we're trying to observe. But that also will lead to some problems, which we will discuss about, which is some of these endogenously tagged proteins are expressed at very low level. It is very hard if you want to perform dynamic imaging experiments. So just a few examples here. I'm not really going to go too much into details into the biology, but what I want to get across, I want to try to show all the different approaches that really benefited from Cell STED. So when I was a postdoc, actually, we started out a project where we wanted to revisit the role of the small GTPase ARF1 in intracellular trafficking. So ARF1, it's a very well-known GTPase, and it has a very known, a very famous role in the formation of COP1 vesicles at the Golgi. And however, when we put a HALO tag directly on the endogenous copy of ARF1 and not just overexpressed ARF1 into the cells, so the overexpressed ARF1 is on the left and the gene-edited ARF1 is on the right, what we found out is that not only it was involved in vesicle formation, but we could really see all these tubular vesicular structures coming out of the Golgi. So this is what the Golgi looks like at the fluorescent microscope in a living cell. It's the nucleus, and that's why we call it the perinuclear area, because I realized that I say that a lot. And these structures are very dynamic. For example, you have a tubule down here that is kind of coming out of the Golgi, and now we know that it attaches to microtubules and moves to the cell periphery. So that was very exciting for us. This is a not very well characterized role of this small GTPase. However, so we set out to kind of try to figure out what are these tubules, which machinery is involved, which cargoes are contained in these tubules. And we kind of realized that this is not something that you can simply do with the confocal microscopy. So this is a confocal microscope. So one of our first questions is clathrin, so like a classical code like clathrin involved? And when we expressed clathrin in the cells, we saw that it is very hard to study dynamics in the perinuclear area, because it is very crowded with vesicles and tubules and endosomes and Golgi. And it's only when we took advantage of Cell STED in this case that we could really appreciate these tubules here, which were coated with little clusters of clathrin all over them. And these are highly dynamic, so they kind of form at the Golgi but then detach, and now we know they move on microtubules and move to the cell periphery. And I just want to stress again that we wouldn't be able to do this work in fixed cells, because the structures are completely disrupted when we fix our cells. So that was my first little example. And another very important thing is that all this machinery, including ARF GTPases, it's all organized on nanoscale sorting platform at the Golgi of around 70 to 100 nanometers in size. And this is a confocal, and this is the STED, where you really see that the machinery is confined to nanodomains at the Golgi. And in the lab, we really want to understand how these nanodomains evolve, which machinery is recruited, which cargo is sorted. And So this is just another example of the kind of experiment we can do. This is endogenously tagged clathrin light chain, which labels the Golgi here. So again, this is the nucleus, this is the Golgi, and we can really start following nanoscale events. For example, this vesicle here forming and then disappearing, and also these nanodomains that are remodeling and fusing and recruiting different machinery at the Golgi. So I really don't want to go into much detail, but this is the kind of experiments you can do. And another very important thing is that just coming back to one of the other points that I made at the beginning is that these recycling endosomes here in magenta and the trans-Golgi network are really packed in the same place of the cells. So if you have a marker for recycling endosome and the Golgi and you image reconfoculated, they kind of sit in this exactly the same area of the cell. So it's hard to even distinguish them. And it's when we do live Cell STED, instead, we can really see that they're completely separated, and we can even appreciate how these two organelles dynamically interact. For example, those tubules that I've showed you earlier that are making contact with the recycling endosomal compartments. So this is another thing we can do. We can look at organelle dynamics and contact sites and how they interact. And actually a questions that I get a lot when I show, that's why I kind of decided to add this next extra few slides here. A question that I get a lot is like, yes, but my protein is bleaching very quickly. I'm only really able to take one frame and then my protein is bleaching. Also, I work with CRISPR knock-ins, which are not very abundant. And my answer to that is that, I mean, there is a little bit of optimization that one can do, but also is that sometimes a single image in a living cell can tell you much more than an image in a fixed cell. And I'm actually going to show you a couple of examples of what I mean for that. First of all, I'm just going to reiterate what I just said. So live Cell STED is really allowing us to take super resolution imaging of fixation sensitive structures. So you wouldn't be able to see all these intricate networks of organelles and tubules without doing live cell super resolution imaging. And my second point requires a little bit more of a kind of detour and explanation. But in one of our projects, we wanted to understand where this protein called ARFGAP1 is. And we came up with an hypothesis, and we thought that ARFGAP1 is actually, we wanted to show that ARFGAP1 is not associated with Golgi vesicles. What I mean, Golgi vesicles, COP1 vesicles. And we did some immunofluorescence imaging, and indeed, we do see that we have an antibody against GAP1 and an antibody against our Golgi vesicles, and we do see that the two markers are not overlapping. However, actually peers have pointed out that these structures that are labeled by ARFGAP1 still look like clusters. They still really look like vesicles. So it's really only when we started using live Cell STED that we could appreciate better the real localization of this protein. So here we have just like, this is a very low abundant protein, so we're not really able to do any like long-term dynamics, but I think one picture says a lot. For example, this is ARFGAP1 here, which doesn't localize to vesicles. So this we now know that this ARFGAP1 is actually continuously labeling the membranes of the Golgi, and it does not localize with vesicles, and it's strikingly different from our vesicular marker here. With our vesicular marker here, which is little tiny vesicles. And so the point that I wanted to make here is that using antibodies and doing immunofluorescence really can give you clustering artifacts and can also mask the true localization of a protein. Instead, if you do live Cell STED, you're really imaging your protein in their native environment. So you avoid clustering artifacts, and you also avoid the loss of resolution that is due to using a primary antibody and then a secondary antibody. And so my take-home messages, or like our lab take-home messages from our experience with live Cell STED, is that live Cell STED is really allowing us to do to follow short-term dynamics like organelles interacting with each other and vesicle budding. And live cell imaging is also required for structures that are sensitive to fixation, like tubular vesicular organelles, and also mitochondria, for example. I have a little bit of experience with imaging mitochondria, but CRISPR is something that you have to use harsh fixation methods, and the Jacobs lab has beautifully shown that using live Cell STED, you can really follow dynamics of CRISPR in the mitochondria. So that's another application, like another cell biological application, for example. And my third point is that with live Cell STED, you have no clustering artifacts or masking, like, or artifacts that are due to performing immunofluorescence experiment and using antibody. And you also have no loss of resolution because your snap tag is just there at no distance from your protein that you want to image, basically. So it really does allow you to do super resolution in the native cellular environment. And something that is on our to-do list, and that was on our to-do list from after the lockdown, is that now that we have an abberior microscope, we really want to play around with all these adaptive illumination strategies like rescue STED to improve our dynamic imaging. And also, the live dyes are on the way, so we're really looking forward to try the live dyes for our experiment. And with that, I'm getting to an end where I want to acknowledge my former colleagues at Yale, so my mentor Jim Rodman, and also Jörg Beversdorf lab, with the people that build all the fancy microscopes that we used, and the people that developed probes and dyes and helped with the image analysis. And I also want to thank my current lab at Freie Universität in Berlin, our two PhD students, Louise and Alex, our technician Ellie, our master and hopefully soon to be PhD students Petya, and our all around helper Stefan. And I thank you for your attention, and I'm happy to take any questions.
Well, great. Those were amazing images and videos. It was really cool to see.
Thank you.
We got quite a lot of questions, so I'll just start with the first one. It's a quick question. Were you using 2D or 3D STED?
We are only really using 2D. Yeah. We are using 3D a lot, but just in fixed cells. In live cells, we try to find like a very flat area of the cell and just image there so that the structures are very two-dimensional and not three-dimensional. But we're actually, Louise in the lab is actually set up like a 3D cell culture so that we can polarize our epithelia into like big 3D acini, which are polarized. And we're using 3D STED a lot in combination with diamine to avoid bleaching. Yeah, there, but only in fixed cells. Yeah.
Okay, thank you. Then next question. Impressive results. Could you elaborate a little bit on the possible frame rates and on which image size they would apply?
Yeah, that's a whole other talk. That's why I kind of don't discuss that, because obviously if you like zoom in smaller, you can go faster, but because you're going faster, your sample bleaches faster, and it's all, I think it's all like a little bit of a trade-off. Like we like to I don't know, can I say something in terms of when you image like a 5 by 5 micron field of view, for example, and you have enough signal, you can go at like 2 frames a second, 1 frame a second in like 2-color mode. But it really all depends from like the amount of signal you have, and it's all much easier when you overexpress protein, and it's all much more difficult when you have endogenously tagged proteins. So it's all, I think you just have to like play around a little bit with the parameters, see what works for you. And also another thing is like what kind of dynamics are you looking at? You know, if you're looking at, for example, if you're looking at our vesicles budding, we like to go at 1 frame per second because that's where we see all the steps. But if you're looking at actin dynamics or microtubule dynamics that are much slower, then you can just afford to take like 1 frame every 30 seconds. So it's all a little bit relative to what you're imaging and how bright your sample is.
So I guess one just has to try.
Yeah.
All right, next question. The ATO 590 is not fluorogenic, so how long do you actually need to wash your samples?
Yes, that's another trade-off that one has to play around with. So ATO 590 is not fluorogenic. If you have a lot of the SNAP and halo, like a lot, like for example, in our overexpressed cells, it was really not a problem. That kind of acts as a sink, and then you don't get as much background somehow. But yeah, we do wash out for like, I would say at least like 1 to 2 hours when we image with that. But for example, I mean, I'm talking about trade-off here because ATO 590 is like a super photostability champion. It's like amazing. It's like super photostable, but at the same time, it's a really high background. So if you have something that is very bright and you can afford to have that little bit of background, then you would go for that. But then if you have something dimmer, for example, what we like to use the 580 rhodamine CP, which I think we figured out that is corresponding to the 590 live and 580 live, which is a little bit dimmer, but also very fluorogenic, so it has less background. So, yeah.
Okay, thanks. I guess it's already our last question now because of time, and it is, when using 2 dyes, 590 and SIR, do you record simultaneously or sequentially?
Yeah, you do line by line sequential, so it's like quasi-simultaneous basically, because the time that it takes to take a line, it's very little, so it's basically like almost simultaneous. But you do have to do 1 line of one color and then 1 line of the other color. Yeah, there is too much overlap between the dyes to be able to just acquire them simultaneously. But it is basically simultaneously because of the very fast line acquisition speed.
Okay, great. I think we have to move on. Thank you again, Francesca. It was an amazing talk. And thanks also for taking the time to answer these questions.
Thank you, guys, for having me.
Sure. All
Although
right. Sorry?
Although we're only a few hours away from each other. Yeah.
That's true. That are the disadvantages and advantages of this time right now, right?
Yes. Thank you very much. I'll stay around. I'm curious to see talks and the demo as well.
Cool. Okay, so as a next speaker, I'm happy to introduce to you my colleague from abberior, Florian Grimm. And I just recently learned that Flo is actually one of the first employees of abberior who already started to work with the company like 8 years ago as a student worker. Flo is a chemist and biochemist by training and is now working on his PhD thesis at abberior, in which he is developing fluorescent and cell-permeable probes. Today, he will talk about membrane-permeable organic fluorophores for life cells and microscopy. Thanks for joining us today, Flo. I'm really looking forward to your talk.
Okay. Thank you, Janina, for your very kind introduction, and welcome everyone to my presentation, which is about the design of organic fluorophores suitable for live cell microscopy, for confocal microscopy as well as for STED microscopy. So the feature and the main feature of these organic dyes is that they can easily pass the cell membrane, entering the cytoplasm of our living cell. We from the abberior company are a team consisting of biologists, biochemists, and chemists, and creating mainly fluorophores suitable for immunostaining. So those fluorophores are labeled to antibodies, and those labeled antibodies are then used for immunostaining experiments. However, since the last 2 years, we have new organic fluorophores in our portfolio suitable for live cell microscopy. And recently, so the last weeks, we have extended this portfolio with new organic dyes, also very highly cell permeable. And during the next minutes and during the next slides, I will show you the design of those organic fluorophores. The reason, why we need fluorescent molecules, doesn't matter if it's organic dyes or fluorescent proteins in microscopy, is of course that in normal microscopy, if you look through our microscope under bright field conditions, what we see is kind of a shadow of our cell and then probably also the nucleus, but then we cannot really distinguish other proteins or structures or even organelles in our cell. To do so, to really distinguish our targets, we have to label those targets with fluorescent molecules, reading out the fluorescent signal by our fluorescent microscope. And then we can create very nice images showing specifically our protein of interest. So in this case, we have labeled TOM20, which is a protein located at the mitochondria. We could also do multicolor staining, for example, and so on and so forth. However, this is a fixed cell, so a cell labeled with antibodies, and therefore what we see is just a momentum. If you really want to see movements, or if you want to observe movements and interactions of proteins, of structures, of organelles, then of course we have to label living cells. The strategies to label living cells are a little bit different. One example is of course the use of fluorescent proteins. This is a very powerful tool in biology, in microbiology, in biochemistry. And what you mainly do is you fuse your protein of interest to your fluorescent protein, creating a fusion protein, and then you can observe your target with fluorescent microscopy. The main drawback of fluorescent proteins is that they are not as photostable as organic dyes. So if it comes to super resolution microscopy, where we mainly use high power laser, then it might be challenging to really create nice images and good images with fluorescent proteins. Therefore, the use of organic fluorophores in super resolution microscopy is favored. So using organic fluorophores for live cell microscopy needs also strategies to label our targets in our living cell. One strategy, for example, is the use of protein tags. The principle is quite the same like with fluorescent proteins. So you have your plasmid consisting of your gene of interest, and right next to it there is a gene for your protein tag. In this case it is SNAP tag, but you can also use halo tag or clip tag or other protein tags. Once you have placed your plasmid inside your cell, then your fusion protein gets expressed, consisting of your protein of interest and your protein tag. The protein tag itself is not fluorescent at all, but it has a reactive site. And if you now enter organic dyes, organic fluorophores, to your cell, and those organic dyes can pass a membrane having a reactive site attached, then your organic fluorophore will react with your protein tag, forming a covalent bond, and then you have indirectly labeled your protein of interest. However, those protein tags are quite large, so they have the same size like fluorescent proteins. And if you have listened to the first talk by Stefan Hell, so in the beginning of July, where he talked about min-flux microscopy, then we know from the physical side there are no limitations anymore to reach resolutions down to 1 or 2 nanometers. The limitations are now on the side of your tag, so you have to choose the right tag. If you have very large and big tags, then you will blow up kind of your actually structure, reducing your resolution. Therefore, another strategy could be also the use of unnatural amino acids. So if you look to this protein or the part of this protein where each of these green bubbles is one natural amino acid, then you could exchange one natural amino acid by an unnatural amino acid. This unnatural amino acid has a bioorthogonal side chain, so a reactive group suitable for click chemistry. And adding now a fluorophore having a tetrazyme, for example, can be used to create a covalent bond under click chemistry, and then you have labeled your protein of interest very specifically with a very, very small tag. Another strategy, and that's the last strategy I will show you, is the use of small molecular ligands. For example, we can use TPP, so triphenylphosphonium salt. This small molecule is not fluorescent, so it's not a fluorophore, but we can add the fluorophore via this carboxylic acid to it, creating a probe suitable to stain mitochondria. So in this scheme, so in this cell, this orange things here are my drawn mitochondria, so I'm sorry, my drawings are not the best, but let's assume we have now labeled with this probe mitochondria. There are also other small molecules available, for example, jasplakinolide, which can be used to label effectins. So if we add via this amine from our lysine side chain fluorophore to it, an organic dye to it, then we created a probe usable to stain actin filaments in our living cell. Another one would be the use of taxols like carboxytaxel, docetaxel, larotaxel. Taxols are mainly used in cancer treatments, and then they will interact with beta-tubulin. If we add also via this amine an organic dye to it, then we create a probe suitable and usable for the staining of tubulin filaments in a living cell. And the last example is the use of Hoechst. Hoechst itself is a fluorophore already, and it will interact with double-stranded DNA, so with the DNA inside the nucleus, for example. However, Hoechst can be, or the absorption maxima of Hoechst is in the UV region of our visible light. So therefore we have to excite our fluorophore with UV light, and this can cause photo damages in our cell. But if we add via this phenyl group an organic fluorophore to it with a redshifted absorption and emission spectra, then we can create a probe where our Hoechst is just the DNA binding domain, so the ligand, and from the other side we have our redshifted organic dye, and therefore we can avoid phototoxic, or let's say light with higher energy, avoiding phototoxicity. But with all these strategies, so it doesn't matter if we use click chemistry or tags or protein tags or the small molecules, we need fluorophores, organic fluorophores, which can enter the cell, which are cell permeable. And then in the last years, it has been shown that rhodamines are quite favored to creating organic fluorophores for being high cell permeable. So the structure of a rhodamine is basically shown here. You have your xanthine moiety, where you have this three-membered heteroaromatic ring system, where X could be, for example, an oxygen or also other atoms. Then you have your amine with different side groups. And on this benzoic ring, you have your carboxylic acid, or 2-carboxylic acid basically. This one is quite important. I will come to this in a second. And another carboxylic acid, or already an amide here, where we can label our ligands or small molecules or reactive group to it, creating all in all a live cell probe. So this organic dye, this rhodamine, can now exist in two different forms. The first I have shown you is the so-called open form, so an form where our fluorophore is fluorescent because we have a fully conjugated pi system. And so in this form our rhodamine has this Twitter ionic character. So we have an anion and a cation, meaning that this fluorophore is less good cell permeable because of the charge. Now with this very important carboxylic acid here, it is possible to form a spirolactone ring via this carbon. So this spirolactone ring now creates an rhodamine being in a so-called closed form, in a non-fluorescent form, and also in a zero-charged form. So we have now a neutral molecule, and this can pass easily the cell membrane, so penetrating through the membrane, reaching the cytoplasm. And there it can exist also in these two different forms, and hopefully will find and interact or react with our target, in our cell, inside our cell. But how can we distinguish whether our fluorophore or rhodamine can exist in an open form or also can exist in the closed form? In this short movie, I will show you the experiment, or the idea of the experiment we can we mainly use to distinguish whether the rhodamine is can exist in an open and closed form. So we have placed this tube above an UV lamp, and we filled this tube with dioxane. Dioxane is a quite unpolar solvent, so with a dielectric constant of 2, and dissolved in dioxane, or we dissolved in this dioxane solution a fluorophore, a rhodamine. And because the environment now is very unpolar, our fluorophore exists only and predominantly in this closed form, and therefore we see no fluorescent signal at all. If we manage it now to exchange the polarity of our environment from unpolar to very polar by, for example, adding water to it, then we see a shift from the closed form to the open form. This can be measured by different dioxane water mixtures, so increasing the water concentration even further, and at a certain point we see an increase of our absorption and fluorescent signal to a kind of a maximum. This can be measured, creating the sigmoidal curve, and now you see at a certain point, so at a certain polarity or also at a certain dielectric constant, we increase the absorption and fluorescent intensity to a maximum. Now at the point where we have reached half of our possible maximum absorption intensity, this is a value we call D50, and this D50 value gives us an idea whether our fluorophore can exist also in this closed form and being therefore highly cell permeable. For example, if we have a fluorophore with a very low D50 value, then it is rather less cell permeable. If we manage it somehow to increase this D50 value, then we also increase the cell permeability of our fluorophore. If we increase the D50 value even further, then of course we end up with the fluorophore being highly cell permeable. At some point, if we increase it too much, then it doesn't mean that our fluorophore is not cell permeable anymore, but it means that our fluorophore will exist only in this closed form, showing no fluorescent signal anymore, and therefore it can be not used for fluorescent microscopy at all. So we need somehow a range where we have to tune our rhodamine. And now I will show you how we can tune our rhodamine being a good cell permeable fluorophore. As an example, I will show you here the tetramethyl rhodamine, or also called TAMRA or TMR. It is a very old fluorophore and kind of easy to synthesize. So TMR has an absorption maxima of 552 nanometers. And as you can see here, a very low D50 value. So it means that it exists mainly in this open form and being therefore less cell permeable. So we as chemists can now change a lot of things at our rhodamine core structure. So we can add directly to the xanthine moiety different groups, or we can exchange at our amines different groups. We can also exchange the oxygen, for example, by other atoms, and so on and so forth. So this will have an effect on the absorption and fluorescence spectra. So it will shift absorption and fluorescence spectra, as well as we will get an increase in our D50 value. For example, if we add electronic withdrawing groups to our fluorophore, to our rhodamine, so adding these halogens, so the fluoroatoms, directly to the xanthine moiety, or adding these 3-fluor ethyl groups to our amines, this will introduce these electronic withdrawing groups and therefore will increase the electrophilic character of this carbon, forcing our molecule to form the spirolactone ring. It means that now, by introducing these electronic withdrawing groups, we will shift our absorption spectra or maxima into the blue region, roughly 50 nanometers into the blue region. And also, we will increase the D50 value drastically. And now we have designed a fluorophore being, or a green emitting fluorophore being highly cell permeable. If we return now back to our tetramethyl rhodamine, and if we look to our oxygen here, what will happen if we exchange the oxygen by one of these group 14 atoms? For example, if we introduce a carbon, then we will shift the absorption and emission spectra into the red region, creating a red emitting fluorophore. And also we will increase the D50 value, having now a red emitting, very nicely and highly cell permeable fluorophore. Or if we go one step down to silicon, creating the silicon rhodamine or silicon containing rhodamine or rhodamine, a very well known fluorophore for life cell microscopy. We create, because we shift the absorption and emission spectra even further to the red region, we will create an infrared emitting fluorophore showing a highly cell permeability because we increased the D50 value drastically. Then there's also another strategy we can use to force our molecule being a good good cell permeable, because we can design or we can synthesize 3 different isomers of one of the same molecule. And this has been shown recently by the chromatin labeling group of Gracidas and Jonas, so it has been published a few weeks ago. And it depends now where we place our carboxylic acid, or later our amide. So either on carbon number 4, carbon number 5, or carbon number 6, creating, as I said, 3 different isomers: isomer 6, isomer 5, and isomer 4. If you look now to isomer number 6, for example, then the D50 value is roughly at 36. So it's quite okay. With isomer 4, we increase it just slightly to 38, 39. But with isomer number 4, we increase our D50 value dramatically to a very high value. The reason now is, and we have to look closer to this part of our molecule, is that this hydrogen here now can form a hydrogen bond between this oxygen. If you look to the 3D structure, then we see that the distance between this hydrogen and this oxygen is shorter in the closed form than in the open form. So in the closed form, we have a distance between hydrogen and oxygen of 1.75 angstrom. In case of the open form, the distance between oxygen and hydrogen is 1.83 angstrom. Means that our spirolactone ring is also stabilized by this hydrogen bond formed between these 2 atoms here. And therefore we increase the D50 value, increasing the cell permeability of our organic fluorophore. With all these strategies, so adding electronic withdrawing groups to it, exchanging the oxygen by a group 14 atom, or choosing the right isomer, so isomer number 4, gives us the possibility to create, to design, to synthesize very highly cell permeable and very high biocompatible organic fluorophores. And with this fluorophores, we need just sub-nanomolar concentrations. And because we need so low or very low value of concentration, we can also reduce the background, even though we don't have to wash our cells anymore. So there are no washing steps needed, and we can avoid background, and we can still avoid the background. And also, because we use just sub-nanomolar concentrations, we will reduce the formation of artifacts. With this, we can now cover quite the full range of our spectra. So, for example, with the very commonly used 488 nanometer excitation laser, we can use a Live 510, for example, which is also usable for STED microscopy when we use the 595 nanometer STED laser. Or with the use of 461 nanometer, we can excite the Live 560 and the Live 590, for example, and with 640 nanometer excitation laser, we can excite Live 610. And these 3 dyes can be used for STED microscopy using the 775 nanometer STED laser. Now I will show you some examples where we stained different kinds of cells. Here we used a fusion protein of TOM20 and SNAP tag, labeling mitochondria or TOM20 inside mitochondria in HeLa cells. So on the left side is the confocal image, where we used Live 560 in combination with our TOM20 SNAP-tag fusion protein. On the right side it is the corresponding STED image. Even though the 560 Live 560 fluorophore is quite far away from our 775 STED laser, we can still use it for STED microscopy. In this case, we labeled the tubulin filaments in human fibroblasts. So we labeled the Live 610 fluorophore to carboxytaxel, a taxol staining tubulin. And if we look a little bit closer into the image, then we can nicely distinguish or nicely see the difference between confocal imaging and STED imaging. If we draw a line profile, then we see each and every single tubulin filament in STED microscopy, which is not possible shown here in gray with confocal microscopy. As I said, live cell microscopy can be used to observe movements. Here we stained double-stranded DNA inside the nucleus. So these are vero cells, or you see the nucleus or the DNA inside the nucleus of vero cells. And this has been stained with Live 590 conjugated to Höchst, creating a DNA probe. And you can observe it over minutes and hours, showing, for example, the movements of the nucleus and the separation of the cell. Also, with Live 560 DNA, we can use this probe not only for live cell microscopy, but also to stain double-stranded DNA inside fixed cells, and not only double-stranded DNA in the nucleus, but also in mitochondria. So we have stained in magenta TOM20 with antibodies, and in green the mitochondria DNA with the probe Live 560 DNA. On the left side the confocal image, and on the right side the corresponding STED image. All images I have shown you are raw data. So with these examples, I really thank you for your attention, and if you have further questions, please mail us or phone us, and we are very happy to answer your questions. Now I'm going to answer at least a few questions, and yeah, thank you again for your attention.
Thanks, Flo. That was an amazing talk, and the images look so pretty.
Yeah, thank you. And thanks
All
for
right.
the organization of this symposium.
Sure, you're welcome. So we got a few questions. First question: what is the optimum D50 value for maximum cell permeability and to be fluorescent to be imaged?
Yeah, so it's not really straight to answer this question. I mean, to this question. So there's, well, for example, the TMR has a very low D50 value, but it is cell permeable. It is less cell permeable. And if you have enough or a very high concentration, you will get your fluorophore or your probe in your cell, kind of. So the yeah, if you manage it to increase it a little bit further, so a little bit higher, then your fluorophore is better cell permeable. So to answer this question, a range of, let's say, it has been shown that a range of 30 to 60, a D50 value of 30 to 60, it's quite good to assume that this fluorophore is a good cell permeable fluorophore. Yeah.
Okay, thanks. Next question. Can a fluorogenic effect upon target binding be observed for the live dyes?
Not in all cases, but there are, for example, live 560. This fluorophore shows fluorogenic behavior. In case of, for example, live 590 and live 610, there is, because we need very low concentrations. So even though we have a fluorogenic behavior or not, it doesn't matter actually, because we need so less, or yeah, just a few little concentration, and therefore we reduce our background signal by using just nanomolar concentration. So yes, there are, for example, live 560 which show fluorogenic behavior, and others are not.
Okay, cool, thanks. So the next question I think fits perfectly. What are the used concentrations with the new dyes, and how long do we need to stain the cells?
Yeah, so normally you can use for cells, you can use 1 to 100 nanomolar concentration, staining concentration. And so if you have, let's say, no time, then you can stain your cell for an hour, and it's okay. So you just need, or you use a little bit more or higher concentrations and less time. Or you can also stain your cells for 24 hours, for example, and using just 2-3 nanomolar staining concentration, which gives you the same effect. And reducing the concentration, staining a little bit longer your cell, is healthier for a cell. So I would recommend to use, as I said, less concentration, but staining a little bit longer. Yeah.
Okay, thanks. Last question. Will the cells actively pump out the probes after a certain time? And if so, how long can then the structures be observed by fluorescence microscopy?
Yeah, so there are cell types which will really pump out effectively with probes. Other cell types like human fibroblasts, for example, they do not, or not that strongly. And what you can do with this fluorophores or with these probes, you do not have to wash your cells. So leave your probes in your imaging solution or in the solution. Even though your cell will pump out the fluorophores, there are still fluorophores in your solution getting inside the cell again. So without washing, you avoid, or kind of avoid, this pump out effect. And also what you can do is you can add verapamil to it, which is an efflux pump inhibitor, and then you also reduce this pump out effect.
Okay, there's one final question I like, so maybe you could just quickly answer it. Would it be possible to use the live dyes in vivo in transgenic animals or in brain slices, for example?
Yes, so we tried it with tissues, so not on cells, but with tissues, and this works. So we stained the DNA in Drosophila organelles, and also the tubulin and actin structures. So yes, that is possible.
Cool. Okay, so that was now really the last question. Thanks a lot, Flo, for joining us, for your amazing talk, and for taking the time to answer the questions.
Thank you.
All right. As our next speakers, I'm now proud to introduce to you my colleagues Martin Meschkat and Isabel Janssen, who are both integral parts of our applications team at abberior Instruments in Göttingen. Martin actually studied neuroscience and did his master's in STED microscopy at the MPE in Göttingen, and after a small detour to electron microscopy during his PhD, where he worked on myelin, he joined our team as an application specialist last year. Isabel is a biochemist by training and did her PhD thesis with Stefan Jacobs at the MPE in Göttingen, where she developed fluorescent proteins suitable for resolved microscopy. In May this year, so actually quite fresh, she joined our team as an application specialist for STED and min-flux microscopy. Martin and Isabel will now show you how to acquire live cell STED images on our facility line microscope. Isabel and Martin, I'm really looking forward to your presentation. The stage is yours.
Welcome to this webinar on adaptive illumination for live cell STED. My name is Martin, and together with my colleague Isabel, we will show you how to use techniques for saving light to obtain great live cell STED data. Imaging living cells is challenging, especially more even so on the nanoscale. Not only are the proteins and labels within the cells moving around, also the cell as a whole is motile and moves during imaging. Additionally, cells are sensitive to higher light doses, which require a good management of the available photon budget per cell and imaging cycle. To do great live cell imaging, you need appropriate live cell labels. These are mostly divided into categories. On the left-hand side, organic fluorophores that bind your structure of interest, and on the right-hand side, genetically encoded fluorescent proteins that are, for example, co-expressed with your protein of interest. Let's look first at the fluorescent proteins available. Fluorescent proteins come in a wide range of colors, starting in the blue-green range with MTFP, followed by GFP variants like eGFP and the highly bright M-neon green, and the more red variants like M-scarlet or M-neptune. As you can see here, the brightest species, and therefore the ones that are most suitable for STED, are in the 500 to 550 nanometer emission wavelength range. One example for STED super resolution on fluorescent protein is the localization of the microtubule binding protein MAP2 labeled with M-citrine as seen on the left. Another protein is FTSZ in dividing bacteria, here visualized with M-neon green in confocal and STED seen on the right. While in confocal no substructure is visible, the use of abberior EZ3D STED enables us to visualize the ring structure of FTSZ in living bacteria. Another example is the visualization of actin in dendritic spines, in this case the redshifted fluorescent protein M-neptune-2. It is especially interesting that these images were acquired in highly complex living tissue, namely the cortex of a living mouse, where the authors were able to measure the diameter of dendritic spines, which is in the range of 85 nanometer. Beside the fluorescent proteins, we have a selection of organic fluorophores at hand, including silicon rhodamine, several abberior live fluorophores in the red or in the orange. These organic fluorophores are very bright and usually more stable than their fluorescent protein counterparts. Also, these dyes come in different emission wavelengths, ranging from silicon rhodamine in the deep red to dyes such as abberior live 580 and Janelia 549 in the orange spectrum, bridging to dyes like abberior live 510 in the green spectrum. So how do we bring these organic fluorophores to your protein of interest? For that we have several options. We either directly link them to certain binding motifs, such as small molecules that bind to actin or tubulin to visualize, for example, cytoskeletal elements, or we use genetically encoded protein tags like SNAP tag or halo tag that are fused to your protein of interest, and then add organic dye molecules that are coupled to the binding partner for the specific tag. One example for a small molecule-based labeling approach is the use of silicon rhodamine that is directly coupled to a microtubule binding molecule like taxol derivates. Here we did STED imaging of mammalian cells labeled with silicon rhodamine tubulin for 2 hours at appropriate temperature using our software-based techniques to reduce the light dosage. Another example is the labeling of DNA using a silicon rhodamine coupled to a Höchst variant, using it to investigate the condensation of DNA throughout the cell cycle. But what can you do if no small molecule binding to your protein of interest is available? You can opt for genetically encoded tags like SNAP tag or halo tag. By designing a vector that includes your protein of interest together with SNAP tag, you can transfect cells and drive the expression of fusion proteins. Then, cell-permeable organic dyes are added that bind to the tag binding element and allow the visualization of your fluorophore with the bright and stable organic fluorophores. One example where the genetically encoded SNAP tag was used is a visualization of mitochondrial cristae as seen in this publication by the group of Professor Stefan Jacobs. Or this two-color approach by Francesca Bottanelli, where mitochondria and endoplasmatic reticulum are labeled by expression of two tagged proteins and adding two organic fluorophores, seen here in green and magenta. They were able to visualize two different biological proteins in living cells reaching 100 nanometer resolution, and were even able to record dynamics as seen here. So much about the labels. What kind of microscope do you need to achieve great live cell STED imaging? First, you need an environmental chamber that provides the right temperature, humidity and gas environment. Then you need a water objective to image your living cells that are immersed in a watery solution. To achieve the best signal to noise, the system should be equipped with a flexible and efficient signal detection and should be optimally aligned. To achieve highest stability in the Z-axis and a smooth continuous imaging, you also want to have a drift stabilization that keeps the image in focus, especially if the environmental chamber is at 37 degrees. As a final tool, you need ways of managing the photon budget to reduce the overall confocal and STED light dose applied to the sample. In this case, we will use an abberior instrument facility line for this imaging session. Here we have different excitation lasers covering the visible spectrum for 405 to 640 nanometer, including an efficient beam scanner and a motorized pinhole as well as motorized mirrors. We also have a live cell chamber installed as well as a spectral detection with a range of usable objectives. This machine can also be upgraded to allow fluorescent time gating, all while being run by an intuitive software platform. On the STED side, the facility line provides multicolor STED imaging in 2D and 3D STED, achieving resolutions below 30 nanometer in XY and below 75 nanometer in Z. We have the adaptive illumination module equipped that allows the minimization of phototoxicity for live cell STED, including a continuous autofocus module to keep the cells in focus. As additional upgrades, the microscope can be equipped with adaptive optics modules for deeper tissue imaging as well as fluorescent lifetime imaging. Also possible is multi-position imaging, allowing for multi-ROI acquisition of several cells per cover slip. The spectral detection we use here can be flexibly adapted between 400 and 800 nanometer, while the highly efficient APDs, avalanche photodiodes, are used to acquire the signal. For live cell STED imaging, we want as much effective light as possible with great STED quality, all while detecting as much signal as possible while minimizing the light exposure time, thereby facilitating sample protection. We achieve this by combining fluorescent time gating with pulse STED lasers to optimize the STED quality, while using the spectral detection for highest detection efficiency. To minimize the light dose, we further apply adaptive illumination. This combination enables us to acquire bright, super-resolved live cell volumes. To achieve even better signal-to-noise with as little light exposure as possible, we use a set of patented technologies called adaptive illumination. These techniques, called rescue and diamond, allow you to minimize the light dose applied to the sample by making sure that STED is only applied to the pixel that have already signaled in the confocal image, thereby preventing the STED illumination of empty image areas that do not contain biological structures. This can reduce the light dosage by over 90% compared to conventional STED. So, how does adaptive illumination work? How do we actively reduce the light dose on the sample? Let's have a look at this model. A biological structure, labeled with fluorophores, is shown in green, and the pixels making up the image are shown as white boxes. A laser beam is used to excite a part of the field of view. As at that position no signal is detected, we do not apply STED in the next step, but we instead just move the excitation laser. As we didn't use any STED, as there's no biological structure in this region, no harm was done to this region or the nearby sample. Now we move the excitation beam a bit further. We start to see fluorescence. What we do next is we do an intermediate STED step. We proceed to STED, but this STED step is just a low power STED probing STED. So this is a fast imaging with limited STED power. This donut already gives us some increase in resolution, but is not applying the full STED power. What we see here is that if we apply a little bit of STED, no fluorescence was detected as we improved the resolution so far, but we saw the structure of interest is not in the center of the excitation beam. We proceed. We move the excitation beam a bit, do again our intermediate low power STED step, but we start to see fluorescence. Now, as we see fluorescence here, we can continue to do high power STED, and here with high power STED we get the maximum increase in resolution. So we use high power STED only where we're really sure there is fluorescence signal with biological structures. As this process is done live during imaging, you can directly visualize the area where no STED is needed, here shown in blue, and where therefore no high power STED was applied, preventing unnecessary STED illumination to the sample. As you can see here, this allows you to image your cells or structures for much longer before the fluorophores are starting to lose their fluorescence. And this allows you to achieve even higher resolution images, as you can apply the highest STED power only to the region of the image where it is really needed. This allows you to use high STED powers to gain maximal resolution, meanwhile minimizing bleaching. In combination with multi-position imaging, this allows you the ongoing imaging of cells at different positions of the cover slip. All this is supported by an intuitive and powerful user interface, enabling a wide range of scientists and students to use these machines to get excellent images. For this webinar, we will now use a fixed nuclear pore sample to establish the use of adaptive illumination schemes, and will then head on to a two-color live cell imaging using abberior live 560 labeling DNA and abberior live 610 labeling tubulin. Now I will hand over to my colleague Isabel for the introduction of the microscope.
Thanks, Martin. Today we will demonstrate adaptive illumination and live cell STED using our facility line microscope. This microscope comes with two interfaces. One for advanced users with an incredible number of imaging options, and the second one we will use today, which is very intuitive and easy to use, and which is especially designed for microscope facilities. Here in the start screen you have the option to do an auto alignment or switch off the microscope, or you can directly go to imaging. In this next window you find these different dye tubes. These represent imaging templates and contain information about the excitation wavelengths, the emission and the stacked wavelengths of the dyes. You can look for a matching dye by text search, so for example the abberior dyes, or by the emission wavelengths using this bar here. I would like to show you the most important functions of the facility line using a fixed sample. Here nuclear core complexes are immunostained with abberior star red. So I take this dye here and name it. This is our fixed sample. And we go for an overview by pressing live. We can now select a cell by just dragging it and focusing as possible using the mouse wheel. I decide for this nucleus here and press live. Now I switch on this resolution switch here and press scan again. So now we already acquire our first STED image. You can adjust the color table here to improve the contrast, and in addition you can also change the color table, for example to a green one. So you see we can have super resolution images in only a few clicks. Next I would like to guide you through the software. In the first row we have the basic options like light path, scan, volume and time for time-lapse. And you can escalate these options and find some settings. In second escalation, you will find even more settings, for example, the spectral detection window. The volume option facilitates easy acquisition of 3D stacks. To show this, I will take new region of interest and press live. And now we can decide for the first plane of our stack and the last plane using these buttons here. I stop it and take the imaging settings from the last image by just dragging it into our new image. So you see the resolution is now switched on again. I briefly adjust the step size to 50 nanometers and the pixel size to 30 nanometer, and then let's scan. Again, we have our confocal image in the left and our STED image in the right. In addition, you find at the top and at the right of the images X, Z and Y, Z projections. Now we can already see in the XY image our improved resolution. But in this mode, we don't improve the resolution in Z. This we can see in the projections. So we have these needle-like structures here in the Z projections. And in the confocal image, we have the these broader structures. So now let's acquire the last images of our stack. And then I take this stack tool here so that we can scroll through our stack. Okay, next I would like to take a new region of interest and press live again. And again I decide for the first plane and the last plane of the stack and take the imaging settings from before. And this time I adjust the pixel size to 40 nanometer and step size also 40 nanometers and go from a 2D to a 3D step beam. So let's scan again and see what will happen. Now you can see already structures in the confocal image but not in the STED image. These are coming now. So this is due to the improved STED sectioning due to our 3D STED donut. The improved Z resolution is also visible in the Z projections. So instead of needle-like structures, we have now these round structures in the projections. Now we are in the second layer of our nucleus. So if this stack comes to an end, we can again scroll through our stacks. So here the confocal one. And now you should see the improved Z sectioning in our 3D STED stack. The last thing I would like to point out at the fixed cell is adaptive illumination. You will find this here. For this I will take a new region of interest again. And I focus a bit. Stop. And now it would like to use rescue. So for this I first take again our STED imaging settings and switch on rescue and adjust the threshold to 5. And we scan. So from this confocal image, now this gray mask is generated. And in the gray areas, the signal was not high enough with the result that the STED beam is not switched on at these positions. So we take our confocal image for the decision between structure and background. So only at positions here in red With sufficient fluorescent signal in the confocal image, we activate the STED beam in our STED image. In this way, we can reduce the light doses needed for our STED image. This is especially beneficial for live cell imaging. So, and with this, I would like to stop the introduction to our microscope so that we can go to the live cells, and I will hand over to Martin.
Thank you, Isabel. Now we switch to living cells. We go for new sample, and as we have the newly launched dyes by our sister company abberior, these dyes are not even yet in our database. So actually what we do is we take similar dyes. In this case we take abberior star red, which has a similar spectrum than abberior live 610 tubulin. And we call this our one color live sample. I click new overview, and first I press live to look for a cell. Indeed, we have some filaments here already, which look quite nice. So I go here to maybe 6%, 7% excitation power, and you can actually see they are already moving. But to prove this, I will take one region here and maybe do a short live cell movie. I go to time. I say I want 4 iterations with 2 seconds interval, and I press scan. So we're getting a confocal movie. Now as this has finished, I will go here to the T-stack tool, and you can see these cells are nicely and happily moving. We now copy this image by dragging it to the overview, activate STED, put it to 15% STED power, and we do again 4 iterations, and I press scan. Same thing here, we get nice iterations, we get a good or very good increase in resolution here, and we will do multiple frames to show you the ease of doing multi-time-lapse imaging in live cell STED. Once this has run through, I will again go to the time module, and we'll have a look at the filaments in time. Now the stack is done. In confocal you clearly see they are moving. Same instead, we see with much improved resolution the movement of tubular filaments in the cell. Now we can not only do 2D, we can actually also do 3D. So I go to an overview and I select a region that is more densely packed, maybe this one. What I do first, I will define a volume. So I want to do a Z stack. I go to volume, to live, and I use my mouse wheel to scroll first up to have the first plane, and then down to have the second lower plane. And I want this to be happening in 50 nanometer Z steps. Then I go to the scan module, I'll set the pixel size to 40 nanometer, and I put the STED to full 3D STED. Activating the STED power here and setting it to 50 nanometer resolution, we will get a 3D STED stack on a living cell. I press start and you see, while in the confocal image all filaments are already visible and no real Z sectioning is taking place, on the right hand side we see nice improved Z sectioning through the 3D STED module. You can actually even see here in the projections, in this case the XZ projection, that the filaments seem to be very bulky here, while here they are independent dots and much thinner. Now this has run through, I'll go through the stack module. You see filaments in basically all planes, and here you have a much better 3D Z sectioning, so showing that these filaments are indeed lying on top of each other and being in independent levels. Now of course STED, while you see the dyes are quite stable, is still a little bit stressful for the cell, so we want to minimize the light dose as much as possible. How can we do that? We use a software-based patented technique called adaptive illumination. As previously discussed, adaptive illumination helps in reducing the light dosage on the sample. We take a new region, let's say we take this one, and I first press live to focus. Let's take this focal plane here, and then I will activate STED, and I will activate adaptive illumination. In this case, I will activate the rescue module. I'll put it to 5%, and I'll press scan and explain what we see. As previously mentioned, rescue works in a way that you do a normal confocal image. You check in the confocal image where there is signal. So here is signal, here is only background. And you want to do STED only when there's enough signal there. So you want to do STED here in these regions and not in these. What happens now? The rescue parameter here defines basically the threshold, what is signal and what not. And if there's not enough signal, like seen here in this mask, it is grayed out, and therefore in the gray area no STED is activated. Only in these areas where it's not gray we activated STED. Meaning this area here is completely spared of STED illumination, and only this relevant area here, the filament, is STED applied. Now we can actually modify this also on the fly. You can say I want to have this value or this threshold to be less stringent or more stringent and modify it, but usually I just set it up in a way that I get a little bit of black around my filaments, so I have nice signal to noise while sparing the cell of at least 50% of the light, because we don't illuminate all these areas, which make up at least, I guess, 50% of the field of view, if even more, I guess. So that's how adaptive illumination works. We can also do a short time frame here, let's say 4 iterations, but this is of course an excellent tool to do long time live cell imaging. And you can see, even despite optimizing and playing around here, the dyes are still fully functional, bright and stable. Now we wait for this to run through, and then we have again a look at this stack here, or at this time stack. And we see here in confocal everything moves fine, but also here in live cell STED the filament's still moving fine and the mask is adapted on the fly, even for moving objects. So this sample does not only contain one color, actually there's a second color for STED in there as well. So I will now close this here, and I will create a new sample. The new sample will contain star red and star orange spectra. This will be used for abberior, live 610 tubulin as before, and this one is abberior live 560 DNA. This is our 2-color sample. I click New Overview, and because I know that these spectra are a little bit different from star orange, I go to the light path module, go to the escalation, and modify here the spectrum a little bit, so I put the detection window a little bit to the left. Then I go to the simple menu here again. I use some excitation, let's say 9% here and 15% here. I press live, and I see signal here already. Now we need to look for a nucleus. So I drag the 2 colors into each other, so we have an overlay, and I suspect the nucleus to be up there. Let's have a look. I drag the cell with my mouse, and I'm looking for a nucleus. There it is. For nicer visualization, I briefly make this nucleus green, so you can clearly see. its structure. Now I select a region of interest. In this case, we select the nucleus. Maybe we select this part here. Actually, let's do the whole nucleus. Then I press live to look for the right plane. So we're actually in the right Z plane for the nucleus and the filaments. You see they are not exactly in the same plane, so I find an intermediate plane to see the STED effect on both. With that done, once this image has run through, I will activate STED and we'll get our two-color STED image. Here on the resolution slider, I put this to 15% STED power for this channel and 15 for this channel, and we'll do another round of scanning. This is now the confocal, and this is the STED as named here in the channel names, and they will overlap then now. I will make star orange here greenish, so it's better in contrast. So this, in this case it's abberior live 560, is now green. And we can clearly see a resolution increase in the red as well as in the green, having super resolved DNA as well as tubular filaments. We now let this run through, and then I will show you how to do multi-position imaging. So you're able to, for example, image 3 independent positions on your cover slip or 3 different cells during live cell imaging, or of course also during fixed cell imaging with ease. You see here clearly the filaments are much thinner and much better resolved, but also the DNA is much better. This now takes a couple of seconds to run through. I can modify the contrast here a little bit to make it look more crisp. So this is now done. I will go to new overview and look for another cell at another position of the cover slip. So I use the microscope controls now to look for another cell. For example, here's one. I drag the nucleus into the center and I will overlay them briefly, select a region of interest here. That's one of the nuclei I want to image in a minute. I go to overview, select another cell. Let's go here. There's another one. I drag it to the center with the mouse. We overlay them again. And here I select the second region. Maybe this nucleus is a little bit turned, so I will turn it as well. And then we actually look for a third cell. Which is hidden here. I press stop, I overlay them, I select a region of interest, and now I have 3 regions of interest from this, and this cell, and now I select these 3 rows with control, go to this time tool up here, the clock, and say I want 10 iterations, so it will image the 3 images one after another 10 times with a certain interval. In this case I want as fast as possible. Now I press start, and what I get in this case is 2-color confocal, but of course you can also do 2-color STED, 1-color STED, volumes, or any other combination you like with these multi-parameter settings here. First we go here, and then we go to the next image, and it will move the stage automatically and position everything correctly, as you can see here. So this will now be running for a couple of minutes. So I would like to conclude the demonstration of the living cells here and now. Thank you for your attention and hope for your questions. Thank you very much.
Great, thanks a lot, Martin and Isabella. for the great demonstration of our microscope. We got a few questions. So the first question is, how long do cells survive STED illumination?
So how long cells survive is basically decided, of course, by the illumination, but also by the environment. So if you have cells that need temperature, that need humidity, that need CO2, and so on, you're of course much better thriving, or they are much better surviving if you have an environmental chamber at 37 degree, and if you have, for example, raw scavenger in there if you want, and then they can survive for quite some time. So there's a Beversdorf paper which basically also tells you how long you can keep these cells going. It depends on the cell line and the environment. The presentation I've shown you before in the actin filaments that were moving around the tubulin, that I imaged for a couple of hours, actually 2.5 hours or so in total. So the cells survive for quite some time. It's a matter of how many frames you do, how large your pixels are, but in general, you can nicely image dynamic processes over a certain time window, and the cells are fine.
Okay, cool. That sounds good. Next question. How about fast processes in living cells? Can those be observed in STED microscopy?
Yes, absolutely. So it depends on the kinetics and the type of the process, but parameters can be adjusted, for example, the scan field and the pixel size to improve the scan rate. And it also strongly depends on the fluorophore. So for a bright fluorescent signal, the dwell times can be reduced to optimize the scan rate even further. And also, photostable fluorophore is very important because this determines the the number of frames to be acquired. For very small objects, it's you can make use of the technique called min field, and here the scan field is reduced to 100 to 200 nanometers, with the result that scanning is, of course, extremely fast. And in addition, this has another advantage because the scan field is smaller than the maxima of the STED donut. The structure of interest does not see the high power STED beam, and with the result that the bleaching is reduced. And here I would also like to stress that, of course, our MinFlux microscope is an alternative option for fast tracking in cells.
Thank you, Isabel. Next question. 2D to 3D STED is a slider rather than a switch. Is there a 2.5D?
Interdimensional thing? No, actually, it's the matter that we can do a 2D donut to get best XY resolution, and we can do a 3D donut to get good XY resolution and best Z resolution. And since these are basically modified on our SLMs and we can use them in the same beam path, we can actually decide how much power we put on the 2D donut and how much we put on the 3D donut. Yeah, so you can practically say, okay, for example, if you want to do 80% 3D, this means you get a very good 3D resolution, not the perfect best, but the very good one, and you get a little bit more improved 2D, so XY resolution. This helps you basically to decide, okay, what I want, what do I want? Do I want only 3D? Do I want only 2D? Do I want a mixture of both? And usually, even if you go to 100% 3D, you still get a 2D improvement. It's just that you can basically direct the power of the STED beam a little bit towards the 2D or 3D donuts. That's what 2.5D would be in this case.
Okay, thanks, Martin. Next question. What is the temperature in the live cell experiment?
Actually, I can take over this as well because I'm sitting here. So usually we use 37 degree in this case as well, because you really want the cells to be as viable as possible. That's also why the filaments moved so fast, because the cells were really at a physiological normal standard. So we use, in this case, 37 degree. But you can also do it at 20 degree. Yeah? That of course slows down your imaging and helps you with very fast processes. But in this case, this was physiological.
Thank you. And now the last question for today. How much slower is imaging with adaptive illumination?
I would like to answer this question. So because the mass is generated from the confocal image in rescue, here it is about 25% slower than a STED-only image. But if you anyway acquire a confocal image with your together with your STED image, then it's not slower at all because the generation of the mass is on the fly.
Okay, cool. Thanks again to both of you for the great demonstration and also for taking the time to answer the questions.
Thank you.
Thank you.
And also thanks to all our speakers for joining us today. So now, I guess that was it. This was the last online Abberior Instruments Summer Symposium 2020. We had actually an amazing time during our summer symposia. I really enjoy looking back to our MinFlux symposium with Nobel laureate Stefan Hell, our STED Paint symposium with Mike Heilemann and Chris Spahn, or our Adaptive Optics STED symposium with Alex Igna and Mary Grace, and of course today's symposium in Live Cell STED microscopy. I, for sure, learned a lot during the presentations, and I hope you did as well. If you missed one of our symposia, no worries, we recorded every one of them, so just contact us and we're happy to send you the link to the recording. And if you have any more questions about STED or MinFlux, or you would like to test our microscopes with your samples, please let us know. So finally, thank you for joining our Abberior Instruments Summer Symposia 2020. And also thank you for your great input and questions. I hope you enjoyed our webinars. Have a nice day to all of you, stay healthy, and bye-bye.